Dear student, I am Dr. Prakash Mungli, a professor in biochemistry. So in this fatty acid oxidation lecture, I will be going over details on beta oxidation of even number carbon saturated fatty acids and also some of the disorders associated with beta oxidation of fatty acids. Let's get into the details. Beta oxidation of even number carbon fatty acids will occur in the matrix of mitochondria. That's the first point that you must remember here in the oxidation of fatty acid. And in the beta oxidation, there will be several spirals of beta oxidation that will be going on, where a fatty acid is oxidized starting from the carboxyl carbon, which is the number 1 carbon. Every beta oxidation spiral, at the end of every beta oxidation spiral, there will be removal of 2 carbons at a time, and that will be removed as acetyl-CoA. Now the fatty acids are shortened by 2 carbon at the end of each beta oxidation spiral, and then it will go into, once it is shortened, it will go back into the next beta oxidation spiral, and the same mechanism will run and 2 more carbons are shortened. Like this, a 16 carbon palmityl-CoA will undergo 7 beta oxidation spiral to release 8 acetyl-CoAs, 7 NADH plus 7H plus molecule and 7 FADH2. Let me show you that here in the figure. Now, as you see, palmityl-CoA, which is a 16 carbon fatty acid. So, number 1 carbon is a carboxyl carbon always, and then we have second carbon and the third carbon. Second carbon is always written as alpha carbon and the third carbon is beta carbon. So in the beta oxidation spiral, this beta carbon is the one that will undergo oxidation, and there will be number 1, 2, 3, 4 steps in beta oxidation spiral, each spiral of beta oxidation. So one spiral of beta oxidation will have 4 steps. At the end of the 4th step, there will be release of an acetyl-CoA molecule, and the fatty acid is shortened by 2 carbon. That means 16 carbon will become 14 carbon plus 2 carbon acetyl-CoA. Acetyl-CoA is a 2 carbon molecule. This 14 carbon fatty acid undergoes similar number 1, 2, 3, 4 reaction in the second beta oxidation spiral, and then again same thing, acetyl-CoA molecule released, and the carbon fatty acid is shortened by 2 more carbon. That means it becomes 12 carbon. Same thing will go on and on. So like this, a beta oxidation of fatty acid, number of spirals will go on, ultimately will break a 16 carbon fatty acid into acetyl-CoA molecules. If you want to calculate how many acetyl-CoA molecules we get from beta oxidation of 16 carbon fatty acid, you simply need to divide that total number of carbon in a fatty acid divided by 12. You get 8 here. 16 divided by 12 is 8. So that means you get 8 acetyl-CoA. Now, in order to get 8 acetyl-CoA, how many spirals, beta oxidation spirals that it takes? If you count like that, if you look at this particular figure here, so this is number 1 beta oxidation spiral. I am going to write that's number 1. And then number 2, second beta oxidation spiral. 14 carbon going in and giving you 12 carbon at the end. So, third beta oxidation spiral, 4, 5, 6 and 7. So total 7 beta oxidation spiral every time. So first spiral to second spiral, it is 14, 14 to 12, 12 to 10, 10 to 8, 8 to 6, 6 to 4, and then 4 breaks into 2 plus 2, that is acetyl-CoA, 2 carbon acetyl-CoA. So 16 carbon, 14 carbon, 12 carbon, 10 carbon, 8 carbon, 6 carbon, 4 carbon breaks into 2 carbon plus 2 carbon. So that means when you count the number of spiral, 16 to 14, that is number 1 spiral, that's number 1. 14 to 12, number 2. 12 to 10, it is number 3, third spiral. 10 to 8, fourth spiral. 8 to 6, fifth spiral. Now 6 to 4, that is 6th spiral. 4 to 2 plus 2, it will be 7th spiral. So total there will be 7 spirals to break 16 carbon fatty acid into 8 acetyl-CoA molecules. And how do you find that? You can't just write it down as 1, 2, 3, 4. We can do it if we have sufficient time in the exam and all that. But the easy way is, once you get get to know 8 acetyl-CoA is there. All you need to do is just subtract number 1 from that 8. So 8 acetyl-CoA, that means 8 minus 1, you get 7, that means 7 spirals. So it takes 7 spirals, 7 beta-oxidation spirals to break 16 carbon fatty acid. So other way what we can do is, so the other way of knowing number of spirals and also number of acetyl-CoA, to get acetyl-CoA numbers, acetyl-CoAs, so what you do is total number of carbon in a fatty acid, here it is 16 carbon, so 16 divided by 2 equals to 8. So 8 acetyl-CoAs. And to know the spirals, all you need to do is 16 divided by 2 minus 1. So that will be 8 minus 1 equals to 7 spirals. Why do we need to know the spirals here? So in each beta oxidation spiral, we are going to get 1 NADH plus H plus and we are going to get 1 FADH2. So that means in 7 spirals, we are going to get 7 NADH and 7 FADH. So we can use NADH and FADH numbers if we know the number of spirals. Okay, this is an important thing that you need to know so that you can calculate net number of ATPs coming from oxidation of even number carbon fatty acid. All you need to do is just change the number of carbon according to the question. If a fatty acid is 16 carbon palmitic acid, 16 divided by 2. If a fatty acid is stearic acid, you know it is 18 carbon, 18 divided by 2. If a fatty acid is 20 carbon, 20 divided by 2. Like that you use the number, change the number according to the type of fatty acid is given there. Now, let me take you into beta oxidation of saturated fatty acid, even number carbon fatty acid. Now, the oxidation of even number carbon saturated fatty acid is shown here in this particular figure. So, as you see, even number carbon fatty acid is shown. It is just taken an example of a general fatty acid, fatty acyl-CoA, number of carbons not mentioned here. We can take palmitic acid, which is the most common fatty acid. Otherwise, just understand the general principles of beta oxidation. Okay, what happens? Always remember a fatty acid which is undergoing oxidation, beta oxidation in the matrix of mitochondria. Always it has to be activated first. It has to have that CoA there. Only activated fatty acid will undergo beta oxidation of fatty acid. Always. So, activation of a fatty acid consumes 2 ATP. Already we have seen that. We need to remember that number. 2 ATPs are already consumed. So, let's see what happens. So, here is the fatty acid. Let's number the carbons first. So, carboxyl carbon is number 1 carbon and the second carbon, third carbon, fourth carbon, and it goes on like that. Always the second carbon is referred as alpha carbon and the third carbon is referred as a beta carbon. So, we are looking at beta oxidation of a fatty acid. So, beta carbon is the one that will undergo oxidation. So, let's remember that point. Now, let's look at the beta carbon. So, the very first reaction that happens in the beta oxidation spiral is catalyzed by acyl-CoA dehydrogenase. This acyl-CoA dehydrogenase is a general name. It can be any type of acyl-CoA dehydrogenase depending on what is that fatty acid there. If it is a palmitic acid or palmityl-CoA, it is a palmityl-CoA dehydrogenase and you just replace all the names down there by palmityl-CoA, right? So, like that, depending on the type of fatty acid, we have all kinds of acyl-CoA dehydrogenases. In fact, all those number 1, 2, 3, 4 reactions, enzymes, we have specific enzymes there, okay. So, that specific enzyme is going to initiate the reaction, that is acyl-CoA dehydrogenase, very first reaction in beta oxidation spiral. So, a specific acyl-CoA dehydrogenase, what it does? It is going to bring in FAD getting into the reaction and will be reduced into FADH2 while your fatty acid is oxidized. So, where is the oxidation? Oxidation is a bond between alpha and beta carbon is oxidized and there is an introduction of double bond there, which I just marked with the green color. highlighted with green color. Now the fatty acid that you got there is a trans, because the bond is in the trans configuration. Where is the bond? Bond is at double bond is at the second carbon, that is alpha carbon, right? So second carbon has a double bond, so that is trans delta 2 fatty enoyl CoA. Enoyl means having a double bond. So that's the fatty acid which is coming out of the first reaction. So that is fatty acyl-CoA is converted into trans delta 2 fatty enoyl CoA. Now this trans delta 2 fatty enoyl CoA will undergo a hydration reaction, and that is catalyzed by, this is an hydration, so there is a water molecule which is added in there. So it is catalyzed by enoyl CoA hydratase, water molecule is added, and it is added, means hydroxyl group is added over the beta carbon. That's where the beta oxidation starts now. So the beta carbon is attached with the hydroxyl group, and that's why the molecule that is coming out as a product here is a beta hydroxyacyl CoA, because beta carbon has got a hydroxyl group. Now the third reaction, catalyzed by beta hydroxyacyl CoA dehydrogenase, oxidation reduction reaction, where NAD is getting into the reaction now. This NAD getting into the reaction reduced into NADH plus, while your beta hydroxyacyl CoA is oxidized into beta ketoacyl CoA. So that means hydroxyl group is converted to keto group at the beta carbon. That's an oxidation there, right? So molecule is, the product that is made is beta ketoacyl CoA, because beta carbon has got a keto group there. Now this beta ketoacyl CoA will be undergoing a thiolase reaction, where CoAsh, that is the reduced pantothenic acid derivative coenzyme A, reduced coenzyme A, entering into the reaction, catalyzed by beta ketothiolase enzyme, and there will be a breakdown of bond between the second carbon and the third carbon, that is alpha carbon, and the beta carbon occurs, and the product that we get here is the acetyl-CoA. So we are going to get acetyl-CoA there, and also rest of the fatty acid will be released as fatty acyl-CoA, and the number of carbons in this fatty acid will be N-2, because 2 carbons are taken out as acetyl-CoA. Rest of the carbons will be N-2 fatty acid. Carbon atoms equals to N-2. And now this fatty acid, which is shortened by 2 carbon, it will go back into the very first reaction to get into another beta oxidation spiral. Now in each beta oxidation spiral, as you see here, there are 4 reactions in each beta oxidation spiral. Number 1 reaction catalyzed by acyl-CoA dehydrogenase. Number 2 reaction catalyzed by enoyl-CoA hydratase. Number 3 reaction catalyzed by beta hydroxyacyl-CoA dehydrogenase. And number 4 reaction catalyzed by beta ketothiolase enzyme. Now number 1, 2, 3, 4, that is 1 beta oxidation spiral, and that means at the end of each beta oxidation spiral we are going to get 1 acetyl-CoA, and the fatty acid is shortened by 2 more carbon. That will go back into beta oxidation spiral again, and this will go on and on until 4 carbons are left over, and the 4th carbon fatty acyl-CoA finally will go into the last beta oxidation spiral, and at the end of 4th reaction, a 4 carbon breaks equal into 2 plus 2 acetyl-CoA. 2 acetyl-CoA, 1 acetyl-CoA 2 carbon, another acetyl-CoA 2 carbon, like this. So even number saturated fatty acid will break into acetyl-CoA molecules. Okay, and this is what is a beta oxidation of saturated fatty acid. Now let's get into some of the important points here, especially the high yield ones, which have got some clinical application and also possible exam questions or something like that. Now number 1 and number 2 reactions in beta oxidation spiral. Look at these enzyme names. Acyl-CoA dehydrogenase. It can be any acyl-CoA dehydrogenase. Long chain acyl-CoA dehydrogenase, medium chain acyl-CoA dehydrogenase, short chain acyl-CoA dehydrogenase, depending on which fatty acid is getting in there, because all kinds of fatty acids, short, medium, long chain fatty acids, as long as they are even carbon, they undergo oxidation here in the beta oxidation process. So we have these specific acyl-CoA dehydrogenases, and this acyl-CoA dehydrogenase in the very first reaction. Now note that in acyl-CoA dehydrogenase reaction, so FAD is the one that is getting into this reaction as a co-enzyme and also a co-substrate here, and the FAD is coming out of the reaction as FADH2, and this FADH2 is going into electron transport chain to give 1.5 ATP. Why? Because FADH2 will enter into complex 2 in the electron transport chain. And as we have studied in the electron transport chain lecture, anything that enters into the complex 2 of the electron transport chain, that is FADH2 entering there, it is going to help in pumping up 6 protons, and that means 1.5 ATPs are generated there. So that means acyl-CoA dehydrogenase enzyme is the one that is making FADH2 in this reaction and allowing this FADH2 go into complex 2 of electron transport chain. Now, how it gets into the complex 2? So, there is a molecule called electron transferring flavoprotein, that is written as ETF. ETF is electron transferring flavoprotein. Electron transferring flavoprotein is the one that will get this FADH2 and give the electrons and protons to coenzyme Q. So this FADH2 will be taken up by, basically FAD to FADH2 happens over electron transferring flavoprotein. That itself is actually the complex 2 in the electron transport chain, because there are 3 complex 2s in the electron transport chain. One of that is this electron transferring flavoprotein, part of the beta oxidation of fatty acid. So FADH2 getting in there, and the electrons and protons are given to coenzyme Q, which will be converted into CoQH2, and then from CoQH2 electrons and protons will be given to complex 3, and like this they will be in the electron transport chain. So protons and electrons will be transported. So the point here is, if there is any defect in complex 2 of electron transport chain, like what if electron transferring flavoprotein is mutated, that can affect acyl-CoA dehydrogenase enzyme reaction in beta oxidation of fatty acid. That's one thing. What if there is a inhibitor of complex 2, like we have carboxyl and tenyl trifluoroacetic acid? These are complex 2 inhibitor. That can also affect complex 2 in the electron transport chain, right? So that can also affect acyl-CoA dehydrogenase enzyme reaction in the beta oxidation. And also note that FAD, it is derived from riboflavin, vitamin riboflavin, vitamin B2. So this vitamin B2 is the one that makes FAD, and the deficiency of vitamin B2, riboflavin deficiency, can also affect acyl-CoA dehydrogenase enzyme catalyzed reaction in beta oxidation of fatty acid. So these are some of the applications, clinical applications that you need to do here for acyl-CoA dehydrogenase catalyzed reaction. And the other thing is a disease that is related with first reaction of beta oxidation is, what if there is a mutation in a gene coding for acyl-CoA dehydrogenase, like a specific type of acyl-CoA dehydrogenase. Example, medium chain acyl-CoA dehydrogenase, medium chain acyl-CoA dehydrogenase. So it is written as MCAD, medium chain acyl-CoA dehydrogenase. So medium chain acyl-CoA dehydrogenase can be deficient, or long chain acyl-CoA dehydrogenase can be deficient. So a deficiency of acyl-CoA dehydrogenase can also occur, and that can lead to defect in the beta oxidation process. So that's another aspect that we can look into. So we can look into acyl-CoA dehydrogenase deficiency, that's one thing. We can look into riboflavin deficiency, that's another application. We can look into transfer of FADH2 into electron transport chain through ETF, that is electron transferring flavoprotein, that's another important point. And also we know 1 FADH2 is going to give 1.5 ATP in the electron transport chain because protons are transported to coenzyme Q and then all the way to complex 4 and helping in pumping up 6 protons. That concept can be connected. and also inhibitor of complex 2 in the electron transport chain like carboxyl, phenyl trifluoroacetic acid. All these concepts can also be tested, and these are the things that you need to know around this particular reaction. Okay, now moving on to the second important reaction here which I have highlighted is beta-hydroxy-acyl-CoA dehydrogenase enzyme. Third reaction in the beta-oxidation spiral. What is the importance of this reaction? So there can be deficiency of beta-hydroxy-acyl-CoA dehydrogenase, especially long chain beta-hydroxy-acyl-CoA dehydrogenase deficiency leading to defect in beta-oxidation. That's one aspect. Second is NAD, which is entering into the reaction. So that NAD, it's a derivative of niacin, and niacin is vitamin B3. So the deficiency of niacin, that is leading to pellagra. So in pellagra there can be decrease in NAD, and decrease in NAD can affect third reaction in beta oxidation of fatty acid, which is affecting activity of beta-hydroxy-acyl-CoA dehydrogenase enzyme. So that is one thing that you must remember. And then NADH plus that is made here, which is worth 2.5 ATP if it enters into electron transport chain. Where it enters into electron transport chain? It enters into complex 1 of electron transport chain. So if there is any inhibitor working on complex 1 of electron transport chain, inhibiting complex 1, so that can also lead to, so that will affect a third reaction in beta oxidation of fatty acid. So like this, so this is how some of the applied aspect or the clinical aspect we can apply to beta oxidation of fatty acid reaction there. Now once we know that application, some of the clinical aspects in these reactions, so now let me tell you where all this NADH and FADH2 produced and how many ATPs that we get from one spiral of beta oxidation. We are going to consider one spiral to calculate ATPs and use that number later. So number 1, 2, 3 and 4 reaction. Out of these 4 reaction as one spiral of beta oxidation. So number 1 reaction gives you FADH2 which is worth 1.5 ATP. Number 3rd reaction gives NADH plus which is worth 2.5 ATP. So that means total we have 4 ATPs coming from there, from the NADH and FADH. And then we get one acetyl-CoA at the end of each beta oxidation spiral from number 1 to number 6. Last spiral we get 2 acetyl-CoA. Let's not take that into consideration. So number 1 to number 6 spirals, at least up to that, we get acetyl-CoA every time at the end of each beta oxidation spiral, and one acetyl-CoA it is 10 ATPs. Okay. Now let's do the totaling. So 4 from NADH and FADH, 10 from one acetyl-CoA equals to 14 ATPs we get from one beta oxidation spiral. So beta oxidation spiral, one beta oxidation spiral will give 14 ATPs. Remember this number. Why we need to remember 14 here? Because every one additional beta oxidation spiral you can simply add 14 while calculating net number of ATPs. Like here, if you know the net number of ATP for 16 carbon palmitic acid, and if I am asking 18 carbon palmitic acid, what is the net number of ATPs that we get upon oxidation? You don't need to go all over and do the calculation. If you know the number for 16 carbon, which is actually 106, all you need to do to get the net number for 18 carbon, it is 106 plus 14 you just need to add, because for 16 carbon fatty acid, so there will be 7 spirals that is going on to give 8 acetyl-CoA, because 16 divided by 2 is 8 acetyl-CoA, 8 minus 1 is 7, so 7 spiral. So for 7 spirals of beta oxidation, a 16 carbon palmitic acid will give 106 ATP. And the question, if it is 18 carbon stearic acid, if the net number is asked, all it is needed is just one additional spiral, because 16 to 18 is 2 carbon extra, that one acetyl-CoA extra is there and it has to be broken down, means separated, and you need one more additional spiral, and in one additional spiral of beta oxidation you get 14 ATPs like what I have just now explained. Okay. So that means 106 plus 14. So, you are going to get 120. That is the net number for 18 carbon stearic acid oxidation. So, if you know the 16 carbon, you can calculate for 18 carbon. Similarly, if your fatty acid that is asked is 14 carbon instead of 18 carbon, if it is asked 14 carbon. So, you know for 16 carbon it is 106. For 14 carbon, all you need to do is 106 minus 14. Minus 14. Because one spiral is less for 14 carbon. So, 106 minus 14. So, whatever the number we get, we get 92. Okay. 92 will be the number for 14 carbon. Like this. So, that's where the understanding of an ATP yield from one spiral of beta oxidation fatty acid is necessary. So, that's what I was trying to say here. Alright, now here is the overview of or the review of the beta oxidation of fatty acid, especially the saturated even number carbon fatty acid from the previous figure that I have explained. So, the very first reaction is acyl-CoA dehydrogenase, where you get FADH2 going into electron transferring flavoprotein, which is actually the complex 2 in the electron transport chain, and you get 1.5 ATPs there. And then enyl-CoA hydratase is the second reaction, and the beta-hydroxy acyl-CoA dehydrogenase is the third reaction, where you get NADH plus, which enters into complex 1, and you get 2.5 ATP there. And beta-ketothiolase reaction, a fatty acid, it is shortened by 2 carbon, releasing acetyl-CoA. And fatty acid equals to N-2 enters into next beta oxidation spiral. Now, this particular slide is showing you how a beta oxidation of palmitic acid is going on and how much energy that we get from upon oxidation, a complete oxidation of palmityl-CoA molecules. So, palmitic acid, which is converted into palmityl-CoA, that's what is the requirement. So, it is a palmitic acid. Let's start from there. Palmitic acid or palmitate, which is a deprotonated form of palmitic acid, activated into palmityl-CoA, minus 2 ATPs there. So, you got to consider that. If the energy is asked for energy coming from palmitic acid, then you will have to take out 2 ATP because CoA addition has consumed 2 ATP. Now, here just for easier understanding, 16 carbon is written, that's 16 carbon here. So, you go one by one now. The very first spiral of beta oxidation, number 1, I will write it in green color, number 1, that's a first spiral. So, you get 1 FADH2, 1 NADH and 1 acetyl-CoA, which is entering into TCA cycle to give 10 ATP. 1 FADH2 give 1.5 and 1 NADH give 2.5 ATP. So, we are going to calculate that total later. Now, like that, 16 carbon is coming converted to 14 carbon. So, going into second beta oxidation spiral, like that 14 to 12, 12 to 10, 10 to 8, 8 to 6, 6 to 4 and 6, 4 to 2. So, like that number 1, number 2, number 3, number 4, number 5, number 6 and number 7. 7 spirals will be going on. Now, the point here is in the 7th spiral of beta oxidation, 7th spiral, 2 acetyl-CoAs will be released instead of 1 acetyl-CoA there, because last spiral 4 carbon gets in and 2 acetyl-CoAs are going to get. So, if you count the number of acetyl-CoAs there, so I am going to mark acetyl-CoAs with the purple color. So, all these are 2 carbons, acetyl-CoAs there. Okay, so total there are 8 acetyl-CoAs, means 8 TCA cycle. As you see here, 8 TCA cycle. Number 1, 2, 3, 4, 5, 6, 7, 8. 8 TCA cycle means 8 times 10. Okay, so that is 80 ATPs, because 8 acetyl-CoAs, each acetyl-CoA is 10 ATP, acetyl-CoA, 8 acetyl-CoAs times 10, so equals to 80 ATP there. And now there are 7 spirals, so it took 7 spiral to break 16 carbon palmitic acid into 8 acetyl-CoAs. So 7 spirals, let's calculate ATP from there. So 7 spiral means 7 NADH plus 7 FADH2. Okay, 7 NADH plus H plus 7 FADH2. 7 NADH will give you 17.5 ATP. 7 FADH2 will give you 10.5 ATP. So if we add all these numbers together, we are going to get 108 ATPs. And this 108 is the total number of ATPs, And if you want to calculate the net ATP yield from oxidation of palmitate, so it will be 108 minus 2. Why? Because 2 ATPs are consumed for the activation of palmitate into palmityl-CoA. So that will be 106 ATP is the net number of ATP that you get from oxidation of palmitate. So this is how a ATP calculation can be done. So if you know the net number of ATP coming from palmitate, which is a 16 carbon, that should be the understanding. Then you use that 14 number. Previously I told you, every spiral give you 14 ATPs. So if the question is 18 carbon oxidation, so it is 106 plus 14 you should add. If it is a 14 carbon fatty acid being asked, it is 106 minus 14. So this is a net number, not a total number. Don't subtract 2 again. So example, if it is a stearic acid that I am asking 18 carbon, so if you are doing 106 plus 14, so you are going to get 120 ATP, and this 120 ATP is the net ATP yield. It's not like total ATP, because 106 is net. You just need to add 14 to get the net number. Don't subtract 2 from that 120 and say go for 118. It is just already it's a net number. All you need to do is, if the question is net number of ATP if it is asked. Okay. If it is asked as a total number of ATP, then you don't need to subtract those 2 ATP. Usually in the questions, net number is asked rather than the total number. Just be careful just in case. If the total number, then you don't need to subtract 108. No ATP subtracted. If it is a net number, 108 minus 2, 106, like that. Okay. So just look at the question and go accordingly. That's about energy from oxidation of fatty acid. Now let's move on to see some of the clinical disorders that are associated with beta oxidation of fatty acid. Apart from, I have explained you riboflavin deficiency affecting first reaction in beta-oxidation spiral, like acetyl-CoA dehydrogenase enzyme being affected in riboflavin deficiency, or in niacin deficiency like pellagra, where NAD is less, affecting beta-hydroxy acyl-CoA dehydrogenase reaction, third reaction in beta-oxidation. Those are the connections with the vitamins. Of course, that can be a clinical application there. Other than that, what are the diseases that are specifically with the beta oxidation? So it is one of the most common beta oxidation defect disorder is a medium chain acyl-CoA dehydrogenase deficiency. So medium chain fatty acyl-CoA dehydrogenase deficiency, simply written as MCAD deficiency. Now, out of all fatty acyl-CoA dehydrogenases which are involved in very first reaction in beta oxidation spiral, so the most common one is medium chain acyl-CoA dehydrogenase deficiency. What can be the other ones? Others are like short chain fatty acyl-CoA dehydrogenase deficiency, long chain fatty acyl-CoA dehydrogenase deficiency. You can write it as SCAD deficiency or LCAD deficiency. Okay, those are also there, but out of all these SCAD, LCAD and MCADs are the most common ones. That's how it is most common out of all acyl-CoA dehydrogenase deficient enzymes. Now it is an autosomal recessive condition like any other genetic conditions. Most of them are autosomal recessive. So there will be impaired oxidation of medium chain fatty acids which are containing 6 carbon to 12 carbons. That's the medium chain there. So because the medium chain fatty acid oxidation is impaired here, that's why patients will show elevated levels of C6 to C12 fatty acids, that is medium chain length fatty acids, and also C6 to C12 acyl-carnitine levels will be elevated because these fatty acids are esterified with the carnitine in the intermembrane space and they become acyl carnitine esters and they accumulate and their levels in the blood will be elevated. That's why you see elevated levels of C6-C12 acyl carnitine, C6-C12 dicarboxylic acids in the blood and urine. And because the beta oxidation is defective here, especially for medium chain fatty acids not undergoing complete oxidation because of the defect in the enzyme. So lack of ATP. So that will decrease ATP levels, decrease in ATP levels because of beta oxidation not completely going on. It's just stopping at the medium chain length, long chain to medium chain, and after that it's not going on. So overall there will be shortage of ATPs in the cell, and that lead to decrease in gluconeogenesis. So gluconeogenesis is the one that will support blood glucose level under fasting condition. So because of the decrease in gluconeogenesis, because of lack of ATP, beta oxidation has to support gluconeogenesis. That's the thing here. So when beta oxidation is defective, gluconeogenesis goes down, ATP level decreases, so glucose synthesis decreases, means patient will go into hypoglycemia. And also because beta oxidation is not completely going on, it is stopping at medium chain carbon length, so not much acetyl-CoAs are coming for ketone body formation. So patient will show no ketone body or low ketone body, no or low ketone body, referred as non-ketotic or hypoketotic hypoglycemia. That's one of the sign that is seen with beta oxidation defect diseases. Any disease that leads to decreased beta oxidation process will show hypoketotic hypoglycemia or non-ketotic hypoglycemia. Now, what is the hallmark of MCAD deficiency? The hallmark of MCAD deficiency is profound hypoglycemia. Profound hypoglycemia. Patient will show low or absent ketone bodies. There will be increase in C6-C12 acyl carnitines and C6-C12 dicarboxylic acids, and also these episodes will be provoked by overnight fast, because overnight fasting will lead to increase in beta oxidation process, and now beta oxidation is defective and that can lead to increased accumulation of these molecules. So, or it can be any infection where beta oxidation is kind of increased but there is a defect here. Now, treatment is IV glucose to prevent hypoglycemia, and frequent feeding and especially feeding of high carbohydrate and low fat diet will help, because we are trying to keep beta oxidation low, thereby decrease the accumulation of acyl carnitine species. Now our next disease in this beta oxidation process is Jamaican vomiting sickness. As the name says, Jamaican vomiting sickness is commonly seen in Jamaica and other East Caribbean islands, and also it is seen in some part of Africa. The reason for this is there is a tree called ackee tree, and this ackee tree bears a fruit called ackee fruit. So ackee fruit is coming from ackee tree, and consumption of unripe ackee fruit. What is the reason for Jamaican vomiting sickness when a person consumes unripe ackee fruit? The reason for that is whenever the ackee fruit ripens, okay, so ackee fruit has like difference between ripe and unripe. So in unripened form, unripe. So in unripe form, there is a toxin which is there in the ackee fruit, and the name of the toxin is hypoglycin A and hypoglycin B. There are two toxins there, hypoglycin A and hypoglycin B. This hypoglycin A and hypoglycin B, out of these two, hypoglycin A is most toxic compared to hypoglycin B. Anyway, both of them will be there in ackee fruit, hypoglycin A and hypoglycin B. Whenever a fruit is unripened, both of them, both hypoglycin A and hypoglycin B will be present in an increased concentration in the flesh of the fruit. It will be there in the flesh. Okay, when it is a unripe fruit, hypoglycin A and B will be there in the flesh. That means whenever unripe fruit is consumed, so hypoglycin A and hypoglycin B gets into the body. Okay. When fruit ripens, what happens? So it is a ripe fruit. Okay. When the fruit ripens, at that time, this hypoglycin A and hypoglycin B will be found in the seeds of the fruit. It will move to the seeds. From the flesh, it will move to seeds. So that means the flesh of the fruit will have less or no hypoglycin A, hypoglycin B. That's in the very less concentration. It doesn't lead to signs and symptoms. So when the fruit ripens, from the flesh, these toxins will move to the seeds and there will be high concentration of hypoglycin A and B in the seeds. Always remember, whether it is a fruit is ripened or unripened, seeds is always poisonous. So the seeds are always poisonous here. Seeds of Aki are always poisonous. Always poisonous because they always contain hypoglycin A and B, whether it is ripe or unripe. But when the fruit ripens, so the hypoglycin A and B, which is also there in the flesh, will move into the seeds. So that means it is safe to consume the flesh of Aki fruit only when it is ripened. That's my point. Now let's see what will happen if a person consumes unripe Aki fruit, especially the flesh. What happens? So toxic hypoglycin A will enter in, which is at high concentration in unripe Aki fruit, will enter into the body, and in the body hypoglycin A is converted into methylene cyclopropyl acetic acid. That's a product of hypoglycin A. And this methylene cyclopropyl acetic acid, which is written as MCPA, what it does, it is going to make a complex with carnitine. You know carnitine is helping in the transport of long chain fatty acids into the matrix of mitochondria, important function. And MCPA will also make a complex with coenzyme A. You know coenzyme A, OASH. And this coenzyme A, it is needed for the activation of a fatty acid, whatever the type of fatty acid it is, short, medium, long chain fatty acids. So you need coenzyme A activation. So the MCPA make a complex with coenzyme A, it makes a complex with carnitine, making an ester there, and thereby, and also moreover, so that thereby it will decrease the availability of free carnitine, free coenzyme A availability decreases because it is all making a complex. Okay, so overall causing deficiency of carnitine, deficiency of coenzyme A, and moreover, apart from that, MCPA is going to inhibit. So MCPA will inhibit several acyl-CoA dehydrogenases also. So there are three effects of MCPA in causation of Jamaican vomiting sickness. It is MCPA is going to make a complex with carnitine. It's going to make a complex with coenzyme A, decreasing their availability, and also it is going to bind with several acyl-CoA dehydrogenases. It can be short chain acetyl-CoA dehydrogenase, medium chain acetyl-CoA dehydrogenases, long chain acetyl-CoA dehydrogenase. It is going to make a complex, means inhibit those enzymes, and these dehydrogenases, acyl-CoA dehydrogenases, they are catalyzing very first reaction in the beta-oxidation spiral. So thereby beta-oxidation of fatty acids are totally decreased. So because of this MCPA action, these three actions, there will be decrease in the fatty acid activation and decrease in the transport of long chain fatty acids across mitochondrial membrane, and also decrease in oxidation of several types of fatty acids. So because of this, what will happen? So overall beta oxidation of fatty acid is down. Oxidation of fatty acid is totally decreased, and the oxidation of fatty acid is decreased. That will lead to decrease in the ATP formation, and when there is a decrease in the ATP formation, some of the processes that are dependent on ATP like gluconeogenesis, so gluconeogenesis will decrease. That's what I have written here. And when the decrease in gluconeogenesis happens, it will lead to hypoglycemia, hypoglycemia, especially when the person needs glucose, like when there is a severe vomiting, dehydration is there, hypoglycemia is setting in because of decrease in gluconeogenesis, right? Or when the person goes into fasting condition, not taking any food, so hypoglycemia sets in and gluconeogenesis is not helping it because gluconeogenesis is down. So this is the pathophysiology of Jamaican vomiting sickness and how hypoglycemia converted to MCPA, which has got so many inhibitory effect on the oxidation of fatty acids, starting from the carnitine there. So here, just for your understanding and to know how this ackee fruit look like, here is the ackee fruit and also the seeds are shown. So seeds are always poisonous, but the flesh of the ackee fruit can be safely consumed only when it is ripened. Now, what are the signs and symptoms of Jamaican vomiting sickness? Sudden onset of vomiting will start 2 to 6 hours after ingestion of unripe ackee fruit dish because of the other components of this unripe ackee fruit induces vomiting, nausea and vomiting, and patient experiences pronounced diaphoresis, that is sweating, tachypnea, increase in the respiratory rate, tachycardia, increase in the heart rate, headache, generalized weakness, paresthesia, that is altered sensation, and disturbed mental status. Unless treatment is given, these episodes are followed by convulsions, coma, and it can lead to death. Okay, so the treatment is admission and then taking out, like stomach wash can be done if still undigested unripe ackee fruit dish is there, and then observation, and importantly give glucose, IV glucose, and monitor the symptoms accordingly. So the death is more common in children than in adults. So that's about Jamaican vomiting sickness. This is all about oxidation of saturated even number carbon fatty acids. So we have seen the steps in the beta oxidation process and the spirals in the beta oxidation process, how to calculate ATPs, net number of ATPs from oxidation of even number carbon fatty acids, and then MCAD deficiency, and Jamaican vomiting sickness. So that's all about beta oxidation of fatty acids and clinical aspects. I hope this video has helped you in understanding this very important concept. Thank you.